Hi everyone, welcome back to Useful Genetics. This is lecture 2G, where we're thinking about finally other causes of mutations than mistakes by DNA polymerase. We'll think about insertions of mobile elements that are a major cause of mutations, and we'll think of some of the consequences, and then we'll discuss changes to chromosomes, duplications and deletions, rearrangements, and changes in chromosome number. Now, we'll start with insertion of a mobile genetic element. And here I've shown the before and after of an insertion of a mobile element, what we call in earlier slides, we referred to as genetic parasite, into a gene. Now, these mobile elements can insert anywhere in the chromosome, but here I've drawn it inserting into a gene. And to give you a sense of how this works, I've made a little animation. So what happens is that a copy of the mobile element comes to the gene and cuts it, and then inserts itself into the gene and seals itself into the gene, so that now we have a continuous DNA double-stranded molecule, part of which is the original gene, and part of which is the new sequence of the genetic parasite. Now, as I said, this can happen not only in genes. If it happens in a gene, it's likely to have very serious effects on the gene's function because it disrupts the coding sequence. Here I've shown insertions accumulating outside of genes, and one way to explain that would be to say that insertions also happen inside genes, but because they're usually destroying the gene's function, natural selection eliminates those mutations from the population. So we don't very often see insertions of mobile elements into genes, but we very often see them elsewhere in the genome. So this is evolutionary time. Initially, a few insertions, and then as time goes on, more insertions of different types of elements. As you saw, the different colors in that pie chart represented different kinds of genetic parasites. And some of these genetic parasites are inserting into sequences that themselves were insertions of genetic parasites. So it's the little fleas have little fleas to bite them. Here's another one where an insertion of a genetic element is inserted inside another one. As time goes on and the genome evolves further, more and more of these elements accumulate. Now we've got pink ones piling up, pink ones inside other ones. The genome is getting bigger and bigger. It's not getting more genes, but it's getting bigger and bigger because it's getting more and more mobile genetic parasite sequences in it. In fact, these mobile genetic, a particular class of these genetic parasites, is thought to have been the original cause of introns, that they began as, They began as insertions of a particular type of genetic parasite into coding genes. And this type of parasite, because it was able to splice itself out of the RNA, didn't destroy the gene, wasn't eliminated by natural selection. And this is how introns are thought to have begun. So I've diagrammed one here. Here's another one where the intron genetic parasite has inserted, and then another genetic parasite is inserted inside the intron. So the introns themselves are also getting bigger because they are also sites where mobile genetic elements can insert into the genome without causing any harm to the function of the gene. Now, this leads us to thinking for a minute about, well, how big are genomes? What determines how big genomes are? So this is a graph that's on the x-axis. It has the size of the genome of many different organisms, viruses, bacteria and archaea, eukaryotes, the size of their genome, and the number of protein-coding genes. Now, we talked about RNA's function of genes, but almost all genes in organisms are protein-coding genes. And what we see is that viruses have very small genomes, Most viruses, a few viruses are very large, but most have very small genomes, but they manage to squeeze in quite a lot of genes, like up here, for instance. Bacteria and archaea have very compact genomes. There's not much junk, not much mobile elements in their genomes, and their number of genes that they contain is quite nicely linearly a function of how big their genomes are. But the situation's very different for eukaryotes. Now we see a whole wide range of genome sizes, from less than 10, almost down to 1 megabase, all the way up to 10,000 megabases. So that's 3 times as big as our genome, and very less dramatic, but still dramatic differences in number of protein coding genes. Here's us, we're this yellow circle. We're actually pretty typical for animals. Our genomes have about 20,000 genes, maybe 30, depending on how you count, and we're about 3,000 megabases of DNA. And most of our genome is not genes. If most of our genome was genes, we'd be over here on the graph. So here's the question. Mobile elements have important consequences. As I said, usually the insertion of a mobile element will eliminate the function of the gene, and that's because if we consider this gene, and for now let's just ignore this little regulatory sequence that I've drawn here, just consider the normal gene, the blue gene with its promoter, ribosome binding site, and start codon. Before the mobile element inserted, this gene encoded protein A. Now that the mobile element has inserted into it, it works in effect as a frameshift. This is a sequence that wasn't selected to be transcribed and translated from these sequences. And so when translation starts at this start, so it will be transcribed into a messenger RNA from its original promoter. But when translation starts from its original start codon, it's going to be translating sequences that are part of the mobile element. And chances are that the protein will be completely non-functional, because these sequences were not evolved to act as proteins, and will truncate prematurely because it'll meet a stop codon. But in the case that I want you to consider, which is actually a fairly common case because many mobile elements contain regulatory signals themselves, I want you to consider the case where the mobile element itself carries a second set of regulatory signals. There's a promoter pointing out of the element, and there's a ribosome binding site and a start codon. And then that's followed by the rest of the blue gene, gene A. And what this question is asking you is, will the protein that's produced from this promoter, from this messenger RNA, will it resemble the normal protein A? And the answer is, well, it could, but it probably won't. Even though in this drawing, almost all of the gene for protein A is intact, and it's got a substitute promoter and binding site and start codon. But there's one problem, which is, what's the probability that we have the correct reading frame here? 2/3 of the time, by chance, 2/3 of the time, we're going to have the wrong reading frame. And so we're going to produce a protein that does not resemble protein A. The protein will be translated, but again, because it's the wrong reading frame, it's likely to hit a stop codon, and the protein will be truncated and non-functional in any case because it's making the wrong amino acids. 1/3 of the time, though, it'll have the right reading frame, will produce what could be a pretty much functional copy of gene A, of protein A, because only a little bit's been removed from the end. And the consequences of this kind of event can then be a change in how gene A is regulated. Instead of being regulated by these signals, now it's regulated by these signals. And that can have very interesting and important evolutionary consequences. And we see these quite a lot when we look closely at the evidence of how gene regulation has evolved. Now, let's switch to thinking about chromosome mutations. And we'll just cover these very briefly because we'll do them in a lot of detail in Module 10. The first kind to think about is duplications or deletions of large segments. We talked about very short duplications and deletions, insertion of a base or two, deletion of a few bases. But larger deletions can have quite dramatic effects. So here's a duplication where this segment shown in red is now present twice. We call it a tandem duplication because the two copies are side by side. So we've still got a copy of the green gene and the pink gene, and we've got a new gene, a hybrid gene, whose first half is of the pink protein and the second half is the green protein. If the reading frames are compatible, this may produce a completely new protein. Here's a drawing of a deletion where the same segment, instead of being duplicated, it's been deleted. And again, we've got a hybrid protein, but at the cost this time of losing our functional copies of both the green and the pink proteins. We can have larger breaks and joins, not within a single chromosome, but between different chromosomes, so that chromosomes can become sort of hybrid chromosomes with parts from different sources. And over long evolutionary periods, we see that quite a bit of this goes on in typical genomes, so that rearrangements of the parts of chromosomes are quite common. Now, finally, we can see changes in chromosome number. For instance, we might lose a whole chromosome due to a mistake in cell division, or in a complementary event, wind up with 3 copies of a chromosome. These events are important both because they change the dosage of genes. So a cell with this genotype would only make half as much of this protein as it normally would, and it would make 50% more of proteins on this chromosome than it normally would. They're also important because they create problems for sexual reproduction. And this will be discussed initially in Module 7 when we talk about sexual reproduction, about meiosis, and then again in more detail in Module 10. So we've talked about 2 kinds of other ways that genomes can change apart from errors by DNA polymerase. We talked about insertions of mobile elements, which are a major factor in mutational change and in genome evolution. We talked about how they happen. They actively insert themselves into other DNA sequences. And we talked about the consequences for gene function and for gene regulation, usually destroying gene function, but sometimes creating new kinds of gene regulation, and the consequences for genome evolution. They're a large part of the reason that our genomes are so big and so full of sequences that aren't genes. And then we talked about chromosomal mutations, about deletions or duplications of segments of chromosomes, breaks and joins that rearrange segments of chromosomes into new combinations, and about changes in chromosome number. Coming up next, we're going to talk about a distinction between 2 kinds of cells that mutations can happen in, germline cells and somatic cells. And this will then prepare us to think about things like the roles of mutations in the development of cancer. I hope to see you there.